Firstly, just to introduce myself, my name is Dan Brown. I'm the chairman of the UK Gaucher Association, and I'm delighted to have been invited to moderate this webinar, which is in relation to the link between GBA, Gaucher mutations, the microbiome and Parkinson's disease. It's an area of significant interest, which has obviously been a huge amount of research in for a number of years now. And I'm sure as many people on this webinar now from both the sort of Gaucher and Parkinson's community, super interested to hear what our panelists have to say. We have 5 panelists on the webinar today, 5 experts, including Professor Darren Hughes and Professor Tony Shapira. So we're going to hear from our 5 experts, and then there'll be an opportunity for a Q&A session at the end. If you do have a question, please press the Q&A button at the top of your screens, plug in your question, and we'll save them till the end and try and get through as many as possible. So I'll move on now to introduce our first speaker, Professor Darren Hughes. Professor Hughes is Professor of Experimental Haematology at the University College London, and as the Clinical Director of Research and Innovation at the Royal Free London NHS Foundation Trust, Professor Hughes has undertaken extensive research into Gaucher disease and is the chair of the European Working Group on Gaucher disease, and is going to be presenting on the latest therapeutic strategies in Gaucher. So I'll hand over now to Darren.
Thanks, Dan. That's wonderful. Just going to share my slides. So it's really lovely to be with you all and to be able to give this talk today. And hopefully you should be able to see my screen with some slides there. So I'm going to talk about some emerging treatments for Gaucher disease, some of which are already in development and others of which are a little bit more theoretical and are not yet in clinical trial. So I'm going to briefly cover these 7 areas. So one of them is the challenges that we have with current treatments. Why do we need new treatments for Gaucher? Some aspects of enzyme therapy and substrate reduction therapy, and then some possibilities for new treatments. And these are things called pharmacological chaperone therapy, heat shock proteins, exosome therapy, and then finally gene therapy, which is relatively near now for Gaucher, so something to watch out for. So for those of you who are aware of Gaucher, either have Gaucher or have been to seminars like this before, you'll understand the basis of Gaucher disease, but I'm just going to mention it here because it's important for understanding how some of the treatments work. So Gaucher is an inherited condition. It's a storage disorder. And by that, it means that various substances within the body that would normally be broken down build up. And that's because the enzyme, which is normally responsible for breaking down this molecule called glucosylceramide, is missing. And therefore, because it's missing, the glucosylceramide builds up in various cells in the body, such as these macrophages. They're immune cells. They build up in the bone marrow, in the bone, in the liver and spleen. And that causes the various problems that we have in Gaucher disease. That leads to this very variable clinical presentation, but it includes bone pain, bone crises, sometimes problems with joints requiring joint replacements. It includes big liver and spleen that can cause discomfort, low blood counts, such as low hemoglobin that makes people tired, or low platelets that cause bleeding. So lots of very different manifestations of the condition, and these can be addressed by the treatments, in fact, that are currently available. But the enzyme therapy, which is the mainstay of our treatment, is given intravenously and goes through the bloodstream to various parts of the body. However, the enzymes are quite big molecules, big substances, and so they can't get to everywhere within the body. And so some of the limitations of our current systemic treatments are that they can't reach all the parts where there are problems. And of course, one of the management challenges is that they can't actually get into the brain. There's something surrounding the brain, which is a protective barrier called the blood-brain barrier. It's very useful because it prevents damage to the brain, but it means that enzyme treatment can't get in there. And in fact, also our current substrate reduction therapy also doesn't effectively get into or stay in the brain. So the brain is currently a difficult area to treat. Bone is also quite difficult. It takes 5 years for the whole skeleton to turn over. And so any damage which has already happened in the bone will take a long time, if ever, to reverse. And there's quite good data suggesting that enzyme treatment and even some of the newer treatments don't get optimally to the cells in the bone which need the treatment. Lungs are also a challenge, and there's a little bit of a lung-blood barrier in the same way there is a blood-brain barrier. So lung problems in Gaucher are relatively rare, but when they happen, they can be quite limiting, causing shortness of breath and difficulties for people getting around. And again, quite difficult to treat with enzyme therapy. And finally, one of the challenges that we have with Gaucher disease are lymph nodes. So lymph nodes are normal glands like tonsils or the glands that people sometimes get when they have an infection, a sore throat. In Gaucher disease, the glands can get swollen with Gaucher cells. And again, this is very difficult for the enzyme to reduce. And although those glands are benign in themselves, they can cause various blockages and various problems for people, which are very difficult to treat. So we're looking at different ways of giving enzyme replacement therapy. So can we give enzyme in a different way, which stays within the body for longer? It's a number of challenges. Can we give enzyme in a way which will actually cross over the blood-brain barrier? Can we give it some kind of a tag which will enable it to mysteriously cross over the blood-brain barrier and get into the brain? Can we avoid the immune response from enzyme replacement therapy? Can we make the enzyme slightly different so that the body doesn't recognize it as something foreign? Can we give it in a nanoparticle or a little vesicle or some kind of even packaged within a red cell so that it goes into the bloodstream and goes into more parts of the body? Those are all potential strategies for getting the enzyme into those 4 areas that I've discussed, which are so far been difficult to treat. Substrate reduction therapy or substrate inhibition therapy is another way of treating people with Gaucher disease. And instead of giving the enzyme to reduce the amount of substrate which is built up, it blocks the synthesis of the substrate. So it prevents the substrate from being made. So instead of breaking down this glucosyl ceramide, the small molecules of substrate reduction therapy block this from being made and building up. Because it's small molecules, it can often be given orally, and tablets not only save people from the some of the difficulties of continuous intravenous treatment, but also the small molecules may penetrate various different parts of the body. And we've seen this with the problems in the lung that some people have, that the new oral treatments might penetrate the lung more effectively than the larger enzymes. So we've seen some good results in that area. So this is the mechanism, just to explain a little bit more. So under a normal circumstances, the level of production of that glucosyl ceramide substrate is approximately equal to its ongoing degradation. But in Gaucher disease, the level of buildup is higher than the normal degradation or breakdown of that substrate. And so therefore we have Gaucher disease. We can break it down either by giving enzyme or preventing that production, so preventing the buildup. That's the overall mechanism of action. There are some different molecules under development for substrate reduction therapy. You will have heard of eligostat, that's the one that's currently available, but there's another molecule called venglustat, which is being trialed for Gaucher disease. I'm going to skip over that one. Venglustat has the advantage of being a small molecule which can penetrate the blood-brain barrier and therefore enter into the brain, and so therefore might help with the breakdown of substrate that is building up in the brain. So it's another way of approaching that area which enzyme can't currently reach. The advantage of venglustat over eligostat, our current substrate reduction therapy, is that it isn't pumped out of the brain. Eligostat, the currently available treatment, can get into the brain, but the brain just throws it out again straight away, it kicks it out, whereas venglustat will stay there. So we're currently running a study at the Royal Free called the LEAP study, and this is looking at the combination of enzyme replacement therapy and substrate reduction therapy with venglustat for patients with neuronopathic Gaucher disease, so the sort of Gaucher disease that affects the brain. And this is looking at the various biomarkers, various molecules that might tell us what's happening in the brain of patients with neuronopathic Gaucher disease, and whether other aspects of the condition improve over the duration of the study. So those are studies which are currently either ongoing or very close to ongoing, or drugs which are currently available. Now, there are some other drugs which are being looked at in other parts of the world which are not yet completely available for Gaucher in the UK. One of them is ambroxol therapy, and this is a sort of chaperone therapy. So chaperones are small molecules which bind to the faulty enzyme and enable it to fold properly. So one of the problems that some people with Gaucher have is that the enzyme is improperly folded, and therefore it builds up in the cells. It can't get to the lysosome where it would normally break down the storage material. So chaperone therapy, due to their ability to bind to the enzyme and move it around the cell, might improve the activity of the enzyme. And again, these are oral, they're small molecules, and they have the potential to cross the blood-brain barrier. Some people in the world are already using ambroxol because it's a drug which has proven to be safe. It's a cough medicine. And there are some people who are trialing it in small groups of Gaucher patients. And in fact, we have been able to give it to 1 or 2 patients in the UK on a named patient basis. But there hasn't been a large clinical trial yet in Gaucher disease. Heat shock proteins are a natural system that help molecules to fold properly and to move around the cell and be more effective. And heat shock proteins guard against the toxicity or the poisoning of cells that sometimes happen when proteins are misfolded. So they're natural chaperones that help cells by helping to refold them and helping proteins to be recovered when the cell in themselves would normally break them down. So we're currently looking at studies of a molecule called arimoclomol. And this is a natural chaperone that will help the cells to make better use of the enzyme which is currently available to them. We're looking at clinical trials to start with that in this next 6 to 12 months. So as a possible completely new mechanism of treatment. So it's been developed to treat Gaucher disease, but because it acts on various other aspects of the cell, as opposed to just the enzyme itself, it might be effective for treating a wide range of storage disorders, not just Gaucher disease. So it can cross the blood-brain barrier and reach the CNS. So again, potentially another mechanism of treating neurological disorders, including neurological Gaucher disease. Exosome therapy is a way of encapsulating enzyme so that the enzyme can get to various areas of the body. And those 4 areas that I talked about, so the bone, the lymph nodes, the lungs, and the brain, again, might all benefit from encapsulating enzymes with exosomes. And that's another area that we're hoping will be set up in clinical trial in the next 12 months or so, and again, potentially beneficial for neurological disease. So finally, I just wanted to mention gene therapy, and this is an important area for us at the Royal Free, and we will be starting gene therapy study for patients with type 1 Gaucher disease during the course of this year, and possibly neuropathic Gaucher disease following on from that. And this is the concept that we can replace the broken genes by injecting the genes into the body. There are 2 ways of doing this. We can either take the DNA, which would normally represent, normally make the enzyme, and we can encapsulate it within a virus. So we use a virus like a little mini spaceship. We put the DNA that is broken, we fix it and put it back into the virus, and then the virus is injected into the patient. It goes to the liver where it secretes the enzyme, and the patient then has a continuous level of enzyme being produced. We have a study using this sort of gene therapy starting later this year. The other way of giving gene therapy is to take cells from the patient and put the missing gene back into those cells, and then to re-inject the cells into the patient, a bit like a stem cell transplant. And we hope that we'll also have an option for doing that as a clinical trial later this year. So you can see this is what it would look like. The virus, which would have the DNA inside it, would go, it's injected through the arm, it goes into the liver, it makes enzyme, and the enzyme therefore is available to degrade the substrate for the patients. And the mechanism of action would be very similar. If we take cells from the bone marrow of the patient, we give the bone marrow cells the DNA, we put those back into the patient, and again, it makes the enzyme that's available to break down the substrate. There we go. So in conclusion, this is actually a really exciting time for development of new treatments for Gaucher disease. I've explained some of the problems with enzyme therapy, mainly around the areas which enzyme can't get to, and some of the areas which are now being developed. So gene therapy, new methods of substrate reduction therapy, and some chaperone therapy. Really happy to take any questions now or later, or for you to contact me if you're interested in any of those studies. So thanks very much.
Thank you, Darylyn. Thank you, that's great. Before we move on to our second presenter, and for any late joiners, just a reminder that we can take Q&A, after presentations, but we're going to try and take them at the end. If anyone does have a question, you can tap the Q&A button on your screen, type, and we'll try and get through as many as we can once everybody is presented. So we'll now move on to our second presenter, who's Professor Tony Shapira. Professor Shapira is a professor of neurology at University College London and head of the Department of Clinical and Movement Neuroscience at the UCL Queen Square Institute of Neurology. Professor Shapira is an expert in Parkinson's disease and is the head of the research group which runs the Rhapsody and Parkinson's disease frontline studies. And he's going to talk to us about the link between Parkinson's disease and Gaucher.
Thank you very much indeed, Dan, for that introduction. And thank you also to Abby for helping organize this event. Let me share my screen with you. So hopefully you can see that.
Yep, we can see that.
Very good. So I'm going to talk just briefly about the link between Gaucher disease and Parkinson's disease. But before I do so, maybe just a couple of explanations about what I'm going to be talking about. So I'm going to refer to GBA, and GBA is the glucose ribosylase gene, and Darylyn has already introduced this, variations or mutations in the gene from each parent will cause Gaucher disease. So somebody who has two abnormal genes will get Gaucher disease. Inheritance from one parent will result in what we call carrier status, where people do not have Gaucher disease. I'm going to refer to GCase. This is the enzyme made by the gene and is responsible for breaking down proteins and other things like lipid in particular. And Darren just introduced the topic of glucosylceramide as the target for the GCase enzyme. And then alpha-synuclein, this may be new to some people in the Gaucher community, but those in the Parkinson community will be familiar with this. Alpha-synuclein is a brain protein, and it builds up in Parkinson's disease, and we believe that it contributes to the nerve cell damage that results in the symptoms of Parkinson's disease. Now, the link between Gaucher and Parkinson's disease probably goes back about 30 years or more, when there were occasional reports, some of which came from Israel, where they have more Gaucher cases, that it seemed that there were more patients than might be expected with Gaucher disease that were getting Parkinson's disease. And interestingly, some of the family members who were carriers without Gaucher disease, but carrier of one abnormal GBA gene, were also getting Parkinson's disease. So over the years, this accumulated so much so that it was a good idea to really collect all of the cases in the world and see if there really was a link between these two disorders. And this was the paper published almost 12 years ago now that really made that link between Gaucher disease and Parkinson's disease. And that's what I'm going to talk about today. So in terms of that link, it's interesting that those people who have Gaucher disease and those who are merely carriers of one GBA mutant gene have a similar risk of Parkinson's disease. So it doesn't matter whether you have one or two abnormal GBA genes, the risk is very similar. And the risk is such that up to about a third of people with one or two GBA gene mutations will develop Parkinson's disease by the age of 80. But that means that 2/3 do not. And this is a point I want to come back to later. If you look at it the other way, at people in the Parkinson's disease clinics, for instance, we find that 10 to 15% of them have GBA mutations. And this is higher in those of Ashkenazi origin, who have a 25% chance of having a GBA mutation if they've got Parkinson's disease. There are particular mutations that are seen more commonly in Parkinson's disease and Gaucher disease. And interestingly, one mutation that I've given as an example here, but there are others, the E326K, that cause or are associated with a Parkinson risk, but not Gaucher disease. Now, what does a patient with a GBA mutation and Parkinson's disease look like? Are they different from all those Parkinson's disease patients who don't have a GBA gene mutation? Well, only very slight differences can be found. For instance, those with a GBA mutation tend to develop their Parkinson's disease if they're going to get it, and remember, only a minority do. But if they do, slightly earlier, maybe by about 5 years. And the rate of progression is slightly faster than those without GBA genes. The pathology is the same, and this is important. So whether or not you have a GBA gene mutation, the root problem is still a buildup of alpha-synuclein. The brain scans of all different types with Parkinson's disease are the same whether you have a GBA mutation or not, and there's the same response to treatment whether you have a GBA mutation or not. Interestingly, those with GBA mutations, but without Parkinson's disease, may exhibit some of what we call the prodromal or lead-up features to Parkinson's disease. And these, for instance, include an impaired sense of smell. There may be very subtle motor deficits, such as a reduction in the speed of tapping. And there may be an increase of what we call RBD. RBD is a sleep disorder, whereby you shout out in your sleep or thrash about in the bed. Depression and constipation are also slightly more common amongst those GBA carriers. And we've been looking at this to see if these people who exhibit all of these, what we call prodromal features, may be more at risk of developing Parkinson's disease than those who won't develop the disorder and won't have these subtle features. So, what is the root problem? Well, we've explained that there's a reduction in the GCase enzyme due to the GBA gene mutation. And a reduction in the enzyme activity leads to a buildup of alpha-synuclein. And interestingly, a buildup of alpha-synuclein also leads to a reduction in the GCase enzyme. So you can see that there's the potential here for developing a self-amplifying cycle initiated by the GBA mutation, causing a reduction in GCase activity, causing an increase in alpha-synuclein, causing a reduction in GCase activity, and so on. And this is what we believe lies at the heart of the link between GBA, Gaucher disease, and Parkinson's disease. Now, Darylyn has given a very nice introduction to the types of therapy that may be useful for Gaucher disease. And it's possible that some of these may be helpful for Parkinson's disease linked to Gaucher disease, or Parkinson's disease that develops in GBA carriers. But remember that many of the drugs that Darylyn is talking about do not enter the brain. And if they don't enter the brain, they're not going to be any good for Parkinson's disease. So if we look at this diagram here, the GBA mutation at the bottom will lead to a reduction in the GCase activity. A reduction in that enzyme activity leads to an increase in the GSLs, or the substrate that Darylyn is talking about. There's also an increase in buildup in alpha-synuclein. So the way in which you can interact in this pathway, there are two that are described here. Up here are the substrate inhibitors, and Darylyn has mentioned these as candidates for treating Gaucher disease, one of which is currently available. Another was recently tried to see if it could slow down Parkinson's disease, but actually did not show any effect. So reduction of substrate for Parkinson's disease is probably not going to be the way to go. The alternatives that are currently being advanced are the chaperones that Darylyn mentioned that increase the GCase activity, and also using genes to increase the enzyme activity by increasing the GBA. So the idea here is that you interfere in this self-amplifying cycle by increasing GCase activity by whatever route you wish to choose. And increasing GCase activity will lead to a reduction in alpha-synuclein, which we hope will slow Parkinson's disease. Now, in the next few minutes, I'm just going to talk about one particular drug that is currently being tried in Gaucher disease, but mainly outside the UK, but is fairly well advanced in terms of looking at it for Parkinson's disease in those that carry a GBA mutation. And this is the drug ambroxol that Darylyn mentioned. Now, ambroxol works in two ways. As Darylyn mentioned, it's what we call a chaperone, which means it can refold the defective GCase enzyme so that it goes into the lysosome. It also has an interesting effect on the genes in the nucleus to increase the gene activity that produces the proteins for the lysosome. So it increases the number of lysosomes. So it works in two ways. It makes sure that the GCase enzyme goes to where it needs to go to work, the lysosome, and it also generally increases lysosomal function. Now, some years ago, we began looking at ambroxol in a number of different models to see if it would increase the GCase activity and, importantly, reduce alpha-synuclein, because if it could do both of these, then it would be a good drug to try in Parkinson's disease. So here's a summary of what has been quite a long period of research, looking at lots of different models with ambroxol. So we started off with skin cells from people with Gaucher disease, carriers of just one gene, and Parkinson's disease, and found that in the laboratory, ambroxol increased GCase activity. The next step was to make a fly model of a GBA mutation. So these flies had introduced into them an abnormal GBA gene. And when this happened, the flies started to lose their dopamine brain cells, which is what happens in Parkinson's disease, and they were not able to fly properly. But when we gave them ambroxol to drink, we found that the enzyme activity in the fly brain increased. They stopped losing their dopamine cells, and they started to fly normally. Then we started to look at human stem cells, again taken from people with Gaucher disease or their carrier relatives or people with Parkinson's disease. And we made human dopaminergic neurons. So these are the brain cells that are most important in Parkinson's disease. And we made these originally from skin cells and made them in the laboratory. And we found that these cells with the GBA mutation had low enzyme activity and high alpha-synuclein, which meant they were mimicking what was going on in the Parkinson brain. When we gave these cells ambroxol, we found the GCase activity increased and the alpha-synuclein fell, which is really important. So we then moved to mice who again had an abnormality of the GBA gene. And we found that ambroxol in these mice increased their brain enzyme activity of GCase and reduced their alpha-synuclein levels. So it seemed as if ambroxol was perfectly positioned to increase GCase activity and reduce alpha-synuclein levels. This then led us to try ambroxol in people with Parkinson's disease, with and without GBA mutations. This was a small study which was published just last year, which looked at 6 months of relatively high dose ambroxol therapy, 1.2 grams. And it's what we call a proof of principle study. We wanted to find out whether the ambroxol in people with Parkinson's disease would get into the brain and increase the GCase protein. We wanted to know if this drug at this dose in these patients was safe and well tolerated over a 6-month period. We wanted to look at the CSF, that's the spinal fluid, to see how much ambroxol got into the spinal fluid and therefore presumably the brain. And we wanted to see if the effects of ambroxol were useful for those with and without a GBA mutation, but with Parkinson's disease. And the results of the study were very encouraging. First of all, the safety of the drug was confirmed. So the drug was well tolerated. There were no what we call serious adverse events. We found that the ambroxol levels in the spinal fluid were 11% of those in the brain, which is really quite high and meant that ambroxol was very good at getting into the brain. We found that the GCase protein level in the spinal fluid was increased by 35%, which is really a very substantial improvement in this defective protein. We also interestingly found that the spinal fluid level of alpha-synuclein was increased. Now you might think, is that really what we want to see? And the answer is yes, because it means it's probably coming out of the brain and into the spinal fluid to be taken out of the body. And finally, we found that the effects were similar on those with and without a mutation, but with Parkinson's disease. So the results are very encouraging. And the next step is to undertake a much larger study to see if ambroxol can slow Parkinson's disease. So this is what we call a phase 3 double-blind randomized placebo-controlled study. And what it's testing is whether ambroxol at the dose of 1.2 grams can slow Parkinson's disease. So we're going to be looking at people with relatively early Parkinson's disease, but on treatment. And we're going to look at those with and without GBA mutations, about 165 in each arm. So it's a fairly large study, 330 people. And we're going to see if what we call the MDS-UPDRS, this is a way in which you can measure Parkinson's disease clinically, is slowed down in terms of its progression in those that receive ambroxol. And there'll be a further 6 months in which everyone on the study will take ambroxol. And there are a number of what we call secondary endpoints. So this study, which is currently in the final stages of planning, will, I think, help to answer the question whether ambroxol has the potential to slow Parkinson's disease in those with and those without GBA mutations. And of course, potentially, if it is effective, we may start to think about making it available to those who are at risk of developing Parkinson's disease and letting them take what is a relatively safe medication, even to prevent the onset of Parkinson's disease. So I will stop there and stop sharing my screen. Thank you very much for your attention.
Thank you, Tony. That was super interesting. I'm going to introduce our next presenter now, Abby Higgins. As Tony said, Abby has organised this webinar, so a big thank you to Abby. Abby is a neuroscience MSc graduate from King's College London, has an interest in the study of neurodegenerative movement disorders and identification of genetic biomarkers of disease. Abby is the current research assistant and study coordinator of the Rhapsody study at UCL. So I'm going to hand over to Abby, who will talk about an overview and an update on the Rhapsody study.
Perfect. Thank you. I'll just quickly share my screen. So I hope everyone can see that. There we go. So yeah, as Dan mentioned, my name's Abby and I am the research assistant on the Rhapsody study at University College London. So some of you will be familiar with the Rhapsody study and might even be enrolled with us, and others may never have heard of us before. So within this talk, I hope there's going to be something for everyone. I'm going to give a general overview of the study. We're going to talk about the rationale and aims behind Rhapsody, a little bit of an update on our recruitment measures so far, the importance of family participation in Rhapsody. Then I'll walk you through our genotyping process and what participation involves within the Rhapsody study. And then if you feel that Rhapsody might be a good fit for you or for one of your family members, at the end of my talk, I'll give you a little bit of information about how you can potentially get involved. So as Professor Shapiro mentioned, within the last 30 years or so, GBA mutations, which we know cause Gaucher disease, have also emerged as some of the most important known genetic risk factors for Parkinson's disease. But what's important to note is that only a small proportion of people with GBA mutations will go on to develop Parkinson's disease. So for us within the Rhapsody study, it wouldn't be very useful to study a small cohort, as we'd have a very small sample size of people who would go on to develop symptoms of Parkinsonism. So the most powerful way that we can study the link between GBA mutations and Parkinson's disease is to follow a large enough group of people with GBA mutations over a long period of time. And that way we can understand why the majority of people don't go on to get Parkinson's disease, but to really look at why the minority does. So within Rhapsody, we look at the early signs and symptoms of Parkinson's disease. And some of the most important features that we study are things like any memory dysfunction that you might be experiencing, something called bradykinesia, which is a general slowing of movements. We also look at any loss of a person's sense of smell. And we also look at things like sleep disturbances that people might be experiencing. So within our work so far, we have 850 participants within the Rhapsody study, which is really fantastic. But in order to get the statistical power that we need to do our analyses and really explore this link between GBA mutations and Parkinson's disease, we're aiming for around 1,000 participants in total, hopefully. And the way that we recruit our participants is that we work with Gaucher disease clinics across the UK. So we're lucky enough to work in Derrylyn's clinic at the Royal Free Hospital, but we also work with other affiliate institutions across the country. But the issue with this recruitment style that we use is that for anyone who has Gaucher's disease or a family member with Gaucher's disease, you're probably well aware that Gaucher's is an incredibly rare disease, and it's estimated that around, I believe, 800 people within the UK have Gaucher's. So even if we got every single person within the UK to sign up to Rhapsody, it still wouldn't be enough for that sort of crucial statistical power that we need within the study. So the other issue that we have is that if we look outside of people who have Gaucher's disease and we want to look at those people with a single GBA mutation, those GBA carriers that Professor Shapiro also mentioned in his talk, unless you have a family history of Gaucher's disease, it's very unlikely that a GBA mutation carrier will know that they actually carry a GBA mutation. And so it's a bit like finding a needle in the haystack. So the way that we try to most efficiently recruit people with GBA mutations is to work with people who have Gaucher's disease. And through them, we can reach out to family members who are much more likely than the general population to be GBA mutation carriers. So to have one single copy of a GBA mutation rather than those two GBA mutations that cause Gaucher's disease. And that's how we can make our numbers up for the Rhapsody study. So to better visualize this kind of pattern of inheritance within a Gaucher disease kindred, I've created this little diagram. And so everyone within their genome has 2 copies of every single gene. And so within this diagram, this top line here with the 2 women actually represents a single person. And so this woman has 1 copy of a GBA with a mutation, as represented by this blue alpha helix. And she has 1 copy of GBA with no mutations. So she is a GBA carrier. Likewise, this represents a man along the side here, and he has a single copy of GBA with a mutation and 1 copy of GBA with no mutations. So if these 2 people were to have children, every single time that they had a child, their child would have a 25% chance of inheriting both the mother's copy of GBA with a mutation and the father's copy of GBA with a mutation, and therefore have Gaucher's disease. On the other hand, every time they had a child, this child would have a 50% chance of inheriting only 1 mutated copy of GBA and 1 copy of GBA with no mutations. So they would either inherit the mother's copy of GBA with a mutation or the father's copy of GBA with a mutation, and then the other copy with no mutation from the other parent. And so these people would be GBA carriers, much like their parents. They wouldn't have Gaucher's disease. And finally, there would be a 25% chance that these two people's children would inherit their copy of GBA without any mutations from their parents, so both from the mother and father, and therefore they wouldn't be a carrier and they wouldn't have Gaucher's disease. And so what we can really see from this diagram is that within a family who are affected by Gaucher's disease, who have a family member with Gaucher's disease, the parents of that person with Gaucher's disease are what we call obligate carriers. So they will definitely have one copy of a GBA mutation. Likewise, their siblings have a 50% chance of also being carriers of a GBA mutation. And this is really why we are very interested in reaching out to the family members of people with Gaucher's disease when we recruit within the Rhapsody study. And so it's anyone who is a blood relative of someone with Gaucher's disease or a GBA carrier. And it's not just true for parent-sibling relationships, but we're also interested in children, nieces, nephews, uncles, aunts, anyone who is over the age of 18 and is a blood relative. Now, I know that Professor Shapiro has already mentioned this, but the slight increase in risk for developing Parkinson's disease is, from what we know, the same for both people who carry a single copy of a GBA mutation and Gaucher's disease. So both of these scenarios are really important within the Rhapsody study and can really help us to understand this link between these genetic mutations and this disease. Something that's important to note about the Rhapsody study, if you are thinking you might be interested in getting involved, but you'd like to do so discreetly, we will never contact a person's family members without their permission. So if you do sign up and you're not sure if you want your family members to get involved, all you have to do is, when we contact you, is just to say, I'd rather not have you contact my family members, or if there are specific family members that you're okay with us contacting, you can specify that as well. But you by no means have to allow us to contact your family. Sorry. So within our genotyping process within the Rhapsody study, within the first year of the study, you will receive a saliva sample kit in the post. And this looks much like the one on the screen at the moment. So what it is it's essentially a little tube that we send to you, and you spit into it, and you post that back to us via a free post envelope that we provide to you. And from this saliva sample submission, we look for two types of genetic mutation. So the first type of genetic mutation that we look for is in the GBA gene. So within the Rhapsody study, we sequence the entire GBA gene for any known variant of GBA mutation, of which there are many, many different mutations, hundreds in fact. And this way we can really identify carriers and identify the specific mutation that a person may or may not carry. And as I mentioned before, we know that these GBA mutations are associated with a Parkinson's disease risk, a slight increase in the risk for Parkinson's disease. And this is actually variable, contingent upon the type of GBA mutation that a person may carry. The second mutation that we look for is in a gene called LRRK2. And we don't sequence the whole of the LRRK2 gene, unlike GBA. We sequence just one particular mutation. And this mutation is called G2019S. And the reason that we look for this particular LRRK2 mutation is that it has an incredibly strong implication for Parkinson's disease. And so we want to make sure that any signs or symptoms of Parkinsonism that we might be seeing within our Rhapsody cohort can't be explained away by the presence of a LRRK2 mutation. What's important to note here is that LRRK2 and GBA are not associated with each other. So just because you have a GBA mutation does not necessarily mean that you'll have a LRRK2 mutation, and vice versa. It's just simply so that we can rule out this possibility. Something to know as a participant or a potential future participant is that within Rhapsody, we believe it's a participant's right to know about their genetic data. And it's completely up to you when you get involved or you sign up whether you know about your genetic results. So when you first sign up to Rhapsody, we go through a consent process, and you are able to specify whether you want to know about your GBA result, whether you want to know about your LRRK2 result, whether you'd rather know about one or the other or neither. And this is quite a flexible process. It's not rigid. So if you decide to sign up and you think, I don't really want to know about this, but then a few years down the line, you actually do want to know about your genetic results, we can go into the portal and we can change those permissions for you. Likewise, when we feed back our positive or negative genetic results, we will call you and we will specify with you that you still want to know about your genetic results before we feed anything back. And that way you still have full control over it, especially if you've signed up a number of years earlier and you're not certain what you said or your mind has changed since then. And we also offer free genetic counselling over the phone that can help you make an informed decision about whether you want to know your genetic results. So all of these resources and regulations are in place to make sure that our participants have full control over this knowledge. So what does participation in Rhapsody involve? So within the first year of Rhapsody, it takes around 45 minutes to an hour to complete. It's completely online. And what it essentially involves is a series of questions about you, anything that might be pertinent to our analysis or pertinent to know, such as a little brief amount of medical history, about any medication that you're taking. We then move on to look at those early signs and symptoms of Parkinson's that we talked about. So we look at the slowness of movement, that bradykinesia that I mentioned earlier, via a keyboard tapping test, where you move between two keys on your keyboard as fast as you can within a certain period of time. We then move on to a series of questionnaires that look at other symptoms, such as sleep disturbances or any anxiety that you might be feeling, things like that. And then finally, we cap this off with a series of cognitive tests. And these cognitive tests are essentially like little video games that we run online. It takes around 10 minutes and it's very intuitive, but it looks at things like memory disruption or dysfunction and alertness or reaction times and stuff like that. Because these cognitive tasks are quite novel, we understand that people maybe might get a little bit confused the first time around that they do these. And so in order to correct for any errors that have come from misunderstanding the tasks, once you complete your cognitive tasks within the first year of Rhapsody, you receive an invite to come back a week later and to repeat these tasks. And like I say, it only takes around 10 minutes to do, but it just means that we're getting some accurate data from you as to how you perform on these particular tasks. So once all of this is done, we then send you out that saliva kit in the post, as I mentioned, with a free post envelope to send back to us. But we also send out a smell test. And this is a series of 4 scratch and sniff booklets that you essentially just scratch off a little panel and you smell it, and then you tell us which of 4 smells you think the panel best represents. And you send that back to us alongside your saliva kit in that free post envelope. And from this, we get a sense of how you function with your sense of smell. And from this first year baseline data, we create the baseline for you that we can say, you've come into the Rhapsody study and this is your performance. And in all subsequent years from that point on, we can refer back to that baseline performance that we have on your file. So in every year after the first year of Rhapsody, we essentially repeat the battery of tests, again, 45 minutes to an hour. What you might notice here is that you don't get another saliva sample kit. That is because unless something goes wrong within our genotyping process, which is very rare but can happen, we will never ask you for another saliva sample submission. You might also notice that we don't ask for another smell test kit in year 2. That's because we send out smell test kits every other year, so you get them in years 1, 3, 5, and 7. So as I mentioned before, Rhapsody is a remote study and can be done completely online from the comfort of your own home. All you need is a computer with a keyboard to do that tap test. You need internet access to access our portal, and you need an email address that we can sign you up with and contact you through. And so if you think you might be interested in getting involved in the Rhapsody study or you'd like some more information, please do visit our website, which is www.rhapsodystudy.com. If you'd like to talk to a member of the team, like a bit more of information, or you might want to refer a family member on to us, please contact us via our email address, which is rhapsody@ucl.ac.uk. If you would like to talk to us over the phone, we are partially working remotely still, so please email us with a convenient number and a time for you and a date, and a member of the team will give you a call back. We also have our social media channels, Twitter, Facebook, Instagram, and that's where we advertise events like this. and also newsletters or anything we think might be pertinent or interesting to our cohort. So that's everything for Rhapsody. The final point that I have to make is that we do have a sister study called PD Frontline, which launched about a year ago now. And this is for people who have Parkinson's disease, but they don't have GBA mutations. They don't have a family history of Gaucher disease or GBA mutations, but they want to get involved. So this is a clinical trials registry. It's online again. You don't need a computer to complete PD Frontline. You can do it from a smartphone or a tablet. It takes 10 minutes to complete, and we have over 1,000 participants at the moment registered with us. And what this essentially will allow for is that in the future, should a person with Parkinson's who's registered within PD Frontline meet the appropriate inclusion criteria for an upcoming clinical trial, they will be able to be contacted through the PD Frontline platform. So it will be a really great research tool, and also good for people who want to get involved in clinical trials in the future. So that's everything from me. So thank you for listening.
That's great. Thank you, Abby. We've got a few questions in the Q&A, but just another reminder that at the end of the 5 presentations, we'll have time to answer some questions. So if you do have a question, please tap the Q&A button at the top of your screen, and you can type in a question, which we can get to at the end. So I'm going to hand over now to our next presenter, Dr. Jane Norton. Dr. Norton is a senior lecturer at the Institute of Liver and Digestive Health, University College London, has a particular research interest in evaluating the role that microbes, such as bacteria in the gut, can play in neurodegenerative diseases such as Parkinson's. So I will now hand over to Dr. McNaughton. I think you might be on mute.
My sincere apologies, the ultimate, faux pas. Well, thank you very much for the opportunity to speak today. As Dan's already said, I have a research interest in the role of the microbiome in disease, and particularly in its role in the evolution of Parkinson's disease. So I'm just going to discuss some of the evidence for that, and a little bit of background of the gut microbiome in the next few minutes. Well, I suspect you already know this, but Parkinson's disease is the second most common neurodegenerative disease. And of course, GBA mutation, which you've heard about today, it is the most common genetic mutation in Parkinson's disease. But an important statistic is that only 30% of patients with the mutation will then go on to develop Parkinson's disease. So a very important question to ask is, why is that? And what are the other factors required to in the patient's conversion to Parkinson's disease? If we can identify those, then perhaps we can identify new treatments and interventions. So for some time now, there has been some evidence suggestive of the role of the gut-brain axis in Parkinson's disease. We know from studies that appendicectomy will reduce your risk of developing PD by 19%, and patients who've had an appendicectomy 30 years before onset will have a later age of onset. Constipation modestly increases the risk of PD by approximately 4%. From laboratory studies, we know that if you inject the synuclein, that is the protein aggregate, into the gut, it migrates up a nerve called the vagus nerve into the brain. And biopsy samples taken from patients who have PD have been shown to have this prion protein alpha-synuclein in the GI tract. But more recently, there has been much stronger evidence for this association. And this has been because of the generation of germ-free mouse models. So essentially, if you have a mouse model of PD, and the mouse has reared in a normal environment, or the mouse is reared in a germ-free environment, you'd see very different things. The mouse that's reared in the germ-free environment will have much lesser features of PD, lesser inflammation in the brain, lesser abnormalities of movement. And this is quite a powerful observation. And if you take that mouse that is germ-free and you give it a stool transplantation from a patient with PD, then it results in full features of PD in that mouse. However, if you give the mouse that had just a normal environment the antibiotics, then their movement problems and the inflammation in the brain is reduced. So these are very strong data to suggest a causal role, at least as a cofactor at least, in the development of Parkinson's disease. Now, people have looked at bacterial studies, and this table is not there for you to see all the information. It's just to highlight the point that many people have looked in the stool, and they have found quite a number of differences. But there's quite a variation in the results, and just knowing which bacteria are more common in patients with PD and which are less common doesn't really tell us information about what's going on in terms of the functionality. So these studies, which have just told us what type of bacteria go up and go down, they're not really very helpful. We need far more information about the bacteria themselves and the products that they produce. So the gut microbiome is a term that you may have come across. The microbiome is the sum total of bacteria and fungi and viruses that inhabit the gastrointestinal system. And it's been termed as the forgotten organ. And I think what's really important to remember now that we have newer technologies is we are understanding a lot more about the gut microbiome, and that it has a functional contribution to our health, but it can also contribute to disease. We know that there are over 100 trillion microbes in our gastrointestinal tract, and the ratio of cells, microbial cells to our human cells is in excess of a ratio of 10 to 1. So they greatly outnumber us in that regard. And in terms of the number of genes, they have over 200 times the number of genes that we have as humans. And so the obvious question is, why are they there and what functionality do they have? And we think of the microbiome as bacteria, but in fact, there are many other different types. There's viruses, there are fungi, and there are even organisms that we haven't fully understood or characterized yet. So there's a lot of information that we are deriving and will derive in the future. Now, a landmark study in this type of research was the Human Microbiome Project. And this was a large study in America, funded to the tune of about 150 million. And they used a lot of very sophisticated technology to look at normal, healthy humans and the different types of microbes in different parts of the body, including the gastrointestinal tract. And they selected them out by their different types of bacteria, fungi, viruses. And what they looked at is all of the genes, not just what type of bacteria they were, but all of the genes within the microbes. But also, importantly, they looked at the products that they make. So telling you what are the sorts of substances that your gut barrier is being exposed to that may contribute to health or contribute to disease. And they divided them up by different sites. So what this diagram here shows you is that all the microbes from the mouth, for example here, oropharynx, they all cluster together. From the gastrointestinal tract, they cluster together. And there are other sites as well. So we have different bacteria, and some of those bacteria prefer certain environments in our body. So this is very helpful to help us understand the functionality. There's a lot of data in linking the microbiome, not just to health, but to disease. And there's a lot of variation in that data. But one overarching principle is that regardless of the disease that you look at, and many studies have been conducted implicating the microbiome in disease, including diabetes, heart disease, liver disease, gastrointestinal disease, high blood pressure, and of course, neurological disease more recently. But the overarching principle is that in health, you have a diverse, not just population, but a diverse functionality of your bacteria. And in disease, you have a reduction in that diversity. And that is a central principle. And as a result of that, some of the treatments that have been that either in use or being studied to alter the microbiome are directed towards increasing the diversity in the bacteria. So prebiotics, probiotics, and even in some instances, transplantation of stool. Another strategy is to actually target the harmful bacteria with antibiotics and another form of treatment via a virus, but that's very much, it's not in current practice. But of course, the more we understand about these bacteria, they themselves, the more we understand from that information, we'll be able to understand newer targets of therapy, and not just that, look at the microbiome to see if it can predict disease progression in individuals that have a susceptibility, that perhaps carrying a GBA mutation, so that newer treatments can be started earlier and which result in a better treatment. So what are the functions of the gut microbiome? So this is an image where you have the gut barrier interface, that's the human side, and these two different types of bacteria that are stuck to the surface. And this is basically what's happening. The bacteria have a very intimate relationship with our gastrointestinal tracts. And it may not surprise you, therefore, that the bacteria help us to, or the microbes help us to induce immune tolerance. So some people have, for example, are intolerant of particular food substances. Perhaps the most common is celiac disease. And so the bacteria are constantly educating the immune system as to what is okay and what is not. And that's particularly relevant in the first 3 years in life. And bacteria, microbes, I should say, they compete with each other. And in health, you have the vast majority of bacteria are, for want of a better term, good bacteria. Bacteria that contribute to our nutritional state, that improve our immune status, and that provide us with nutrients. But there are a small number of bad bacteria that can cause disease and make us sick. And when you have fewer bacteria, you have fewer bacteria keeping those bad guys in check. So having a diverse population is really important, so those bad bacteria don't overgrow and cause disease. The bacteria will also ferment fibers. So we take fibers and, they're not ordinarily digestible, but the bacteria, particularly in the right part of your large bowel, can produce substances that help, that can be used as an energy, as a food, as we like, for the cells that line the gut. And that's very important. They produce vitamins, particularly folic acid. And they also play a role in the regulation of cholesterol via a substance called bile acids. So they have some very, very important functions, and they well deserve the title of an organ. And we are now understanding more about this. But also we know that there are a number of factors that can influence the microbiome composition. And when we're designing studies, we have to really take these into account so that we can try and adjust for it. And the most profound influence on the composition of the microbiome is diet. Okay, so your a combination of the range of different foods that you eat, but also, for example, vegetarian versus vegan versus omnivorous. You know, these are important distinctions. Drugs can impact not just antibiotics, but also drugs like antacid tablets, such as proton pump inhibitors, because they inhibit the acid barrier in the lining of the stomach, which is there to sterilize the contents of what you eat. So these are important that we make an information, and also some of the drugs that are required for the management of Parkinson's disease can influence bacteria composition. Alcohol has a large effect, but also age and gut motility are important. So when we do research, we like to collect this information. So just thinking about the gut-brain axis more broadly, the neural connection between the gut barrier here and the brain, and this is the area particularly affected in Parkinson's disease, is a circuit which involves a specialist cell called an enteroendocrine cell that is distributed throughout the gastrointestinal tract. It makes a connection with this nerve, which is called the vagus nerve, which goes up to the area in what we call the midbrain, and can influence how healthy the nerves are. And this cell receives signals from gut bacteria and also from nutrients. One of the main purposes of this is to coordinate our responses to having had a meal, but also to sense the bacteria. And so there is a hypothesis called the Brach Hypothesis, which states that the alpha-synuclein, which is the little protein that aggregates and in Parkinson's disease, collects here in the enteroendocrine cell, sticks together, forms these fibrils, which then go up these nerves and go into the midbrain and cause damage and cell death. Sorry, apologies. So some levels of evidence that have shown us or provided evidence for this hypothesis is that if you inject these toxic fibrils into the gut barrier, then they go up the vagus nerve and they cause disease. But if you if you cut this nerve, the vagus nerve, called a vagotomy, then you prevent that process from happening. And so that, therefore, taking all this together, the key question is the microbiome driving this process in which the aggregates or the fibrils are going up into the brain? At this point, I just want to qualify that this is not the only route in which the alpha-synuclein can cause disease in the brain, but there is considerable interest now in this route. And of course, what you've already heard is that the brain is an organ which is very difficult to penetrate. It's very difficult to administer drugs into, but the gut is very straightforward. And we already have many tools, but there are many other ways in which we can perhaps stop this process from happening. And so this is the key question that we hope, one of the key questions that we hope to answer within the body of the Rhapsody study. And I want to just hand over now to Elisa, who's going to explain how we're going to collect some of this information within the Rhapsody study.
Right. Thank you, Dr. Norton. So, as Dr. Norton said, we'll hand over to Lisa Minozzi, who's a neurologist currently undertaking PhD at University College London. She's interested in evaluating the microbiome as a potential risk factor for developing Parkinson's in people carrying the GBA mutations which cause Gaucher disease. So I'll hand over to you.
Thank you so much. Can you confirm that you can see my screen?
Yes, we can, yeah.
Perfect. Okay. First, thanks very much, Dan, for the introduction, and thank to all the speakers who have so nicely and clearly presented these data about the link between the microbiota, Parkinson's disease, and GBA. You have definitely helped my job today. What I would like to do today is, in fact, to present you a new research project led by Professor Shapira and the Rhapsody team and supported by UCL and ASAP, which is indeed focusing on the very interesting link between the microbiota, Parkinson's disease, and GBA. And before going into some more details related to the project, such as the research questions behind the project, who can participate, how you can get involved, and what the study involves, and ultimately why this is important and what we would like to achieve with this project, I would just very, very briefly summarize what has been discussed so far, so what we know and we do not know about the link between the microbiota and Parkinson's disease and GBA. Starting with the microbiome and Parkinson's disease, as Dr. Norton has explained to us, we do know that the microbiota has a very important role in our health because it controls so many processes. The microbes in our body can break down fibers and other components of food. They can produce vitamins. They control the immune system. They protect us from infections. So they are really, really important. And we know that people with Parkinson's disease have different microbes compared to people without the disease. And from the animal studies that Dr. Norton has shown, we also know that the presence of specific microbes in the gut can promote the progression to Parkinson's disease. However, what we do not know yet is whether the presence of specific microbes can also promote the progression to Parkinson's disease in a human subject. And we also know that these gut microbes can produce a huge variety of substances and metabolites that are very important for our organism, but we do not know exactly which metabolites these microbes produce, and especially how these metabolites can affect the intestinal and the neurological function in people with Parkinson's disease and healthy controls. And what about Parkinson's disease and GBA? We have heard from Professor Shapira and Abby that having a GBA mutation is indeed a risk factor for Parkinson's disease, but only a minority of people who have a GBA mutation, 10 up to 30%, will develop Parkinson's during their lifetime. So what we don't know is what are the factors that can promote the progression to Parkinson's in a small proportion of GBA carriers. And on the other hand, what are the factors that conversely protect the majority of GBA carriers from developing Parkinson's disease? So the main question, which is behind our new project, is indeed what are, and if there are, some specific microbes that can increase the risk of developing Parkinson's disease in people who carry a mutation in the GBA gene. And more specifically, what are the, we can say, bad microbes that can promote the progression to Parkinson's disease in people with the GBA mutation? And on the other hand, what are the good microbes that conversely protect people from developing Parkinson's disease? And ultimately, what are the metabolites, the chemicals, the substances that these microbes produce? How we're going to achieve this? Of course, we need your participation, and every participant in Rhapsody is more than welcome to join this project. In particular, we are interested in evaluating the microbiome of people who carry a GBA mutation but do not have Parkinson's disease, people who have a GBA mutation and also have Parkinson's disease, as well as people who have received a diagnosis of Parkinson's disease but do not carry a mutation in the GBA gene. And we also need household controls, so people who live with people with GBA mutation or people with Parkinson's disease and who do not have GBA mutation and do not have Parkinson's disease. A member of our staff might contact you in the forthcoming weeks to propose you to participate to the study and to give you more information if you want. But if you belong to any of these groups and you think that this project might interest you, you want to have some more information, please get in touch with us. We will be more than happy to give all the details that you need. And what exactly this study involves? Well, this is quite simple. So the first step, we will send you a saliva and a stool collection kit in the post at home. If you are a Rhapsody participant, as Abby has explained, you might be familiar with this kind of kit. You will receive, in this case, 2 kits, one for saliva and one for stool collection, with all the instructions to collect the samples. It's very easy. And once you have collected them, you can easily return them in the free envelope that we will send you as well. And then we will also ask you to join us and come to the Royal Free Hospital to perform a full clinical evaluation. We want to look a little bit more in detail into your motor function, so do some motor tests, some cognitive tests, ask you some more questions. In particular, we are interested in having some answers from you about your lifestyle habits, your food, what you prefer to eat, what medication are you on, your bowel movements, because as Dr. Meg Norton has said, these are very, very important factors to consider when we evaluate the microbiome. And finally, we might also ask you to donate some blood and urine samples when you come to the clinic. And then after 1 year, we will send you at home another saliva and stool collection kit to repeat the collection from home and send back the kit to us. And what we are interested in looking at? Well, essentially, we want to evaluate both the types, the species of microbes that are more or less represented in people who carry a GBA mutation, but also the type of metabolites of substances that these microbes produce. And we will compare the data that we get from people who have a GBA mutation to the other groups that I've mentioned before. And we will follow up you after 1 year time to see whether over the time there is any change in the microbes composition which can be associated with the development of those typical symptoms that we see in people with Parkinson's disease, such as slowness of movement or bradykinesia, as Abby mentioned. And we will also compare the metabolites assessment in different samples, so stool, blood, and urine. And of course, we will analyze whether the presence of certain types of food or the use of certain medications is associated with specific type of microbes. Why this is important and matters? Well, because we believe that the assessment of the microbiome might become an important marker of Parkinson's disease risk in people who carry a GBA mutation. And since we know that the microbiota is mostly determined by environmental factors that we can potentially control and manipulate, such as the food that we eat or the medication we take, we believe that in the long term, we might be able to address people who have a GBA mutation to interventions that can target and change their microbiota and therefore prevent the development of Parkinson's disease. And with this, I've finished my presentation. I thank you all for your attention and your participation. And again, if you want to get in touch, if you want to have some more details or information about the project, please write us. This is our email address, and we will be more than happy to be in contact with you. And I'm happy to take any questions. Thank you.
Thank you, Dr. Menozzi. Okay, so we actually have quite a lot of questions now. We'll get through as many as we can. I think we have sort of 15 to 20 minutes. I think any that we don't get to answer, a list of answers and questions for the session can be made available afterwards to everyone who's attended, to the audience. So I think there was some general queries about or comments from people saying that they would be willing to participate in research projects. So maybe after the event, maybe details of all of the research projects could be provided to the audience, Abby, if that's possible, so we can deal with it that way. There was also some general Q&A about if somebody has a relative or somebody close to them who seems to be showing symptoms of say Parkinson's, who may be unwilling to get medical attention, what would be the best way of dealing with it? So I don't know if maybe Professor Hughes or Professor Shapiro might want to just quickly comment on that before we go into the detailed questions.
Maybe if they're showing features of Parkinson's disease, I'll take that, and any answer is they certainly should seek medical attention. Initially, of course, to their general practitioner who can make an assessment, and then perhaps, if necessary, referral to a neurologist. The reason why this is helpful is that people often suffer the symptoms of Parkinson's disease for some years before they get treatment, and that impairs their quality of life very significantly. And having seen so many Parkinson patients like this over the years, as soon as they start treatment to make them feel better, they all say, I wish I'd done this so many years before. So it's really important for their own quality of life to seek medical attention and get it treated. And of course, now that we're starting to look at treatments that may slow Parkinson's disease down, the earlier people will start that, the better.
Thank you. Okay, so we'll turn to some of the specific Q&A. Some of the questions are quite technical, so I'll do my best to get the pronunciation of some of the words right. So this is probably a question for the Parkinson's specialists on the webinar. So the question is Parkinson's disease a prion disease? The precautions taken with other prion diseases are quite severe. Should similar precautions be taken with PWP? Are the Parkinson's plus syndromes similar to Parkinson's disease in this respect? i.e. are Parkinson plus syndromes and MSA prion diseases? That might need some translation, I suspect, for some of the audience.
Okay, so maybe I'll take that again, as it's a question on Parkinson's disease specifically. So the question really is about whether Parkinson's disease is a prion disease, and people on the audience may remember prion diseases were associated with mad cow disease and other disorders. Parkinson's disease, MSA, and other related disorders are not prion diseases in that respect. The reason why the word prion has been associated with Parkinson's disease is because abnormal alpha-synuclein can spread from one cell to another and induce changes in the way that alpha-synuclein folds. So in that way, it can spread from one brain cell to another. But this is not prion disease. It's not infective. It's not infective from one patient to another, one person to another. So in that respect, it's very different. So no precautions are required that might be necessary, for instance, with a prion disease.
Okay, thank you. So I think maybe another question for you, Professor Shapiro. Two questions here related. Could you just touch again on what you said about potential faster progression for, I think the question is for people with the GBA mutation in relation to Parkinson's, and if Ambroxol is used to hopefully slow down progression, sort of by how much, at what rate, and what different symptoms perhaps have a different reaction to treatment?
So people with GBA mutations and Parkinson's disease do progress slightly faster, but you can only tell this by looking at hundreds and hundreds of patients with and without GBA mutations and comparing them. For each individual patient, it really makes hardly any difference at all that you can tell. And in terms of Ambroxol, how much will it slow it down? Well, in the clinical trial, we're looking at it slowing it by just a bit over 18 months. But you can imagine that if you can slow it down just a bit over 18 months, over 10 or 20 years, you'll be slowing it down an awful lot. So the intention with Ambroxol is to see if it can slow down even a little bit. And what will it slow? Well, we hope that it will slow what we call the motor symptoms, so the slowness and the stiffness. But because Ambroxol goes all over the body, we also think it will slow down the progression or development of some of the non-motor problems as well. So we're hoping that if it works, and we have to prove that yet, it will affect many of the symptoms with Parkinson's disease and improve them over time.
Thank you. So the next question, maybe one for Abby. Someone's asked if you could explain the relative chances of a relative having the GBA mutation other than siblings, so the sort of wider relative group that you mentioned would be helpful for the Rhapsody study.
Yeah, absolutely. So it really depends on whether you carry a single or a double copy of a GBA mutation. So for example, if you have Gaucher's disease, any child that you have will automatically carry a single copy of a GBA mutation that they will inherit from you. And then, of course, whether your partner also has GBA mutations as well will affect whether that child will inherit a second copy or not. So whether they'll be a carrier or will also have Gaucher's disease. And so, yeah, that's so for parent-child relationships, again, it's the same as the reverse of what we were talking about. In terms of other relationships, so for example, nieces and nephews and such. So it really depends on the grandparents, what mutations they have, whether they had one or two, and how these were passed on to their children. And therefore, your niece or your nephew will obviously, it will be relevant to whether your brother or sister had a GBA mutation that they can pass on. So it kind of filters out that way. It's very specific to whether you have a single mutation or not, or what your parents had. So.
Thank you, Abby. Maybe move on to some questions directed at Dr. Norton. Are there differences between microbiomes in Parkinson's disease patients and non-Parkinson's disease persons? If so, are these different causes for or consequences of Parkinson's disease?
Well, it's a very good question, and a very astute question, because of course slow intestinal transit is a feature of later disease, and this is why we're trying to conduct a longitudinal analysis and also to have patients with different control groups and at different stages of disease to see if we can disentangle the causal from the consequential. But there is data suggesting that some patients who have prodromal features of Parkinson's disease, they do have a disordered microbiome, and some of the data that I've presented does suggest a causal link. In practice, not everybody, it's a bit like the genetic predisposition doesn't result in a complete, it's not a complete guarantee that you'll get disease. In fact, it's very low, only 30%. So there will be some people for whom well, it's the data suggests, and what our hypothesis is that the gut microbiome will contribute to the disease pathogenesis, but there is data, concrete data, that patients with established Parkinson's disease have altered microbiome. So, in summary, prodromal features, so before the development of movement disorder and once established movement disorder, both have altered microbiomes. But of course, we want to try and understand what's different about the alteration of the microbiome in the early stages compared to the later stages, because that's where we can hope to intervene.
Thank you. Next question is in relation to sort of age ranges of those with the GBA mutation that are also presented with Parkinson's. I think there was a statistic shown around percentage of those with the GBA mutation who have Parkinson's by the age of 80. The question is, what's the actual range of ages as a percentage of those with both Parkinson's and the GBA mutation?
So maybe I'll take that again. So the reason why we quote the figure at age 80 is that the development of Parkinson's disease is age-related, whether you have a GBA mutation or not. So by age 60, really quite a small proportion of people with a GBA mutation, much less than 30%, will get or develop Parkinson's disease. So the figures are much lower the younger you are. So that's why we give as an indication for age 80. As this is the time, probably the maximum time by which someone will develop Parkinson's disease. So the top figure is somewhere between 10 and 30%. And the reason why there is that variation is that it does vary a little bit between different populations around the world.
Thank you. There's lots of questions here about Parkinson's. I'm trying to spread the questions around across the different topics. If a patient with Gaucher disease is at high risk of Parkinson's disease, can anything be done to prevent developments or catch it earlier so it can be treated? Obviously, these trials and these studies are intended to do that, but anything else that more generally can be done?
Not at the moment, and this is what we're working very hard to try and do, to try and identify people who are at risk. And to find something that will slow down those that have Parkinson's and prevent it in those that do not yet have it, but will be at high risk. So that's what the Brach study is about. It's what Rhapsody and PD Frontline are all about, trying to get to this situation where we can identify those at risk, prevent the disease, or slow it down.
Thank you. Another one for Dr. McNaughton. How does good gut bacteria versus poor gut bacteria, how is that determined?
Okay, I'm sorry, it's a term that's loosely used, but there are some key features. So probably the best way to think about it is a healthy microbiome is a diverse microbiome, okay? So that you have a lot of representation. The good bacteria, you often hear that in adverts for probiotics and the like, and they are good. The reason that they're good is that they help to produce nutrients which are useful for your gut health essentially. The bad bacteria are bacteria that normally exist in your bowel, but are very, very low percentages. And what keeps them in check are high populations of the good bacteria. So, but when you have either antibiotics or other factors in the gut that lowers the diversity, these guys spring into action, and they have a whole raft of different mechanisms by which they can be more deleterious. So you don't need to. So the bad bacteria are normally less than 5% of your normal bacterial population, and the good bacteria kind of everyone else. As a very oversimplistic way of understanding it. And the things that contribute to a diverse gut microbiome are a broad diet, a healthy broad diet, limited use of certain drugs that can reduce your bacterial diversity. And although there is no proof, and I saw a comment about probiotics, there's no, it's very difficult to find a positive study in probiotics because everyone's so very different. But in principle, probiotics are there to augment your bacterial diversity, to help restore it with the sort of bacteria that are useful in your gut. So I can't say as a doctor that there's an evidence-based proof, but we know in Parkinson's disease that there is a reduction in bacterial diversity. So probiotics are useful in principle.
Thank you very much. I think we've probably got time for one more question, and I'll just repeat what I said earlier, that I think any questions that haven't been answered will try and be answered after the event, and the questions and answers will be distributed to anyone who's attended. So maybe one final question. Can peripheral neuropathy lead to Parkinson's?
Okay, that's a short answer. No.
Maybe for the lay people on the call like myself, you could just explain peripheral neuropathy.
Yeah, so peripheral neuropathy is where the small nerves in your, particularly your feet, but also eventually in your hands, malfunction, and you get like numbness, tingling, weakness, usually beginning in the feet, but then developing the hands. Many things cause peripheral neuropathy. Things like probably the commonest cause is diabetes. But does peripheral neuropathy cause Parkinson's disease? Definitely not.
Okay, thank you. I think that's all we've got time for today. So firstly, thank you to the audience for joining us for the event. There is a recording being made of this webinar, which will be made available on YouTube. And if you've got any queries about the webinar, please direct those to the Rhapsody email address, rhapsody@ucl.ac.uk. And finally, just a huge thank you to our 5 panelists, Professor Hughes, Professor Shapira, Abby, Dr. McNaughton, and Dr. Minosi. And thank you again, Abby, for organising the webinar, which I think has been super interesting. And I'm sure there'll be lots of questions. I can see more popping in as I finish up. So as I said, we'll do our best to answer everyone's questions afterwards. Thanks, everybody.
Thank you.